The immune system is designed to protect us from foreign material and foreign pathogens. In this video, we are going to talk about specifically the first line of defense in the immune system. Before we talk about this in detail, we'll talk about the 3 lines of defense and the characteristics of each of them individually. Now, though the immune system works together, we can oftentimes break it down into what we call the first, second, and third lines of defense. The first and second lines make up what we call the innate immune system, whereas the third line makes up what we call the adaptive immune response. In the first line of defense, we have barriers, both physical and chemical. Examples of physical barriers can include skin, mucous membranes, and secretions such as tears. The goal of these physical barriers is to basically prevent a microorganism from gaining entry into the host. Similarly, chemical barriers function in the same way, to prevent microorganisms from gaining entry into a host or from being able to colonize in a host. Examples of chemical barriers can include antimicrobial peptides that the body produces, acids such as those that are found within the gastrointestinal tract or the stomach, and also lysozyme, an enzyme that we've talked about previously that'll break down the cell wall of gram-positive bacteria. The second line of defense is more cellular. In this case here, we have white blood cells, for example, macrophages and neutrophils, which are both defined as phagocytes, that are going to basically gobble up or eat up pathogens. We also see the use of cytokines, which are cell signaling molecules used to regulate the function of cells in the innate and adaptive immune system. Now, in the third line of defense, we see the use of B cells and T cells and also antibodies, and we'll focus on this in greater detail down the road. It's important to note here that there are some differences between the innate and the adaptive immune system. And again, we talk about them separately, but remember that they work together to control the immune response of an individual. So the innate immune system is oftentimes thought of as being general. It recognizes patterns in pathogens rather than specific features, which is what the adaptive immune response identifies. Secondly, the innate immune response is immediately active. It's always primed and ready to go in a healthy individual. The adaptive immune response may not be immediately active. In fact, it takes a few days for this to kick in. However, the adaptive immune response has memory. And so if our body sees the same pathogen a second time, essentially, we think of the adaptive immune response as having memorized or remembered that pathogen. The innate immune response has amnesia. It doesn't matter if it's seen the pathogen 10 times or only 1 time. It will respond to that pathogen the same way every single time. Now, it would be a disservice to you if I didn't take at least a minute to talk about the cells that get generated that are part of the innate immune response and the adaptive immune response. Blood cells are originated from what we call a multipotent hematopoietic stem cell. Multipotent means that it can differentiate into different cell types. Hematopoietic tells us that these cell types are going to be blood cells in nature. And these stem cells originate from the bone marrow. So based on the signals that these cells receive during the development, they can develop into a group of cells that become part of the innate immune response, including megakaryocytes, basophils, neutrophils, eosinophils, the 2 monocytes, macrophages, and dendritic cells, and also natural killer cells are part of the innate immune response. And we'll talk about the role of each of these different cell types, or at least the majority of them, over the next few lectures. Now, there are those also from the lymphoid stem cells that develop into those that are going to be part of the adaptive immune response, and these are going to include our T lymphocytes and B lymphocytes. So long as an individual has these hematopoietic stem cells, they are capable of replenishing all of these different cell types that are shown in this picture. In future lessons, we'll also talk about this other part of the immune system, which is known as the lymphatic system. The lymphatic system is a way for different fluids to be passed through locations where B cells and T cells are going to be waiting to come into contact with pathogens. If you've ever had swollen lymph nodes, which oftentimes can occur under the armpits, or sometimes these occur within the neck region, right under the jaw, these are usually signs of infection. And so we'll talk about this system in greater detail down the road. But for right now, I'd like you to know it exists. Let's talk a little bit more in detail about the two first lines of defense, that being physical barriers and chemical barriers. In this image here, I am showing you examples of physical barriers. For example, on the left-hand side, what we see is a cross-section of skin with underlying layers. Well, it's important to note that on the top of this, we have keratinized dead cells that are resistant to breakdown. These are not going to be prone to degradation by bacteria. In addition, these keratinized cells are going to be shed from time to time, which is also going to mechanically remove bacteria with them. Now, we should also note here that within this physical barrier, we do also have chemical barriers. For example, fatty acids that are produced by us or also by bacteria that may reside on our skin, may have inhibitory effects on bacterial growth. Salts or that dry, arid environment can also play a role in selecting against or in favor of certain bacteria growing. And then we also produce antimicrobial peptides, which may be found on these outer surfaces as well. Now, if we focus in on cells in a little bit more detail, what we find is that our cells are kind of held together by what we call cell junctions. And these can come in the form of tight junction, desmosomes, or gap junctions. Tight junctions are kind of like rivets that hold together cells very tightly together. Desmosomes kind of function like shoelaces, as you can see in this picture here, to hold together cells and allow them to potentially communicate from each other. And that is also what we see with gap junctions as well, that they allow for fluids to be exchanged and maybe some information or different chemicals to be exchanged from cells as well. However, in each of these cases here, they provide impenetrable barriers that prevent microorganisms from getting in between those cells. Now, in addition, we could also think of our natural microbiome as being part of a physical barrier as well. The microorganisms that line our gastrointestinal tract or colonize our gastrointestinal tract prevent potentially pathogenic microorganisms from colonizing these GI tracts. And so just the physical presence of microorganisms can create competition that prevents other microorganisms from colonizing these locations. It's also important to note that these microorganisms that are resident to us are also going to be producing short-chain fatty acids and other products that may inhibit the growth of other microorganisms. Now let's take a minute or so to talk about the mucociliary escalator, one system that I really enjoy discussing because of its potential effects on health and how it can be affected by also environment but also by genetics. So in this very poor rendition of a human being here, I've drawn in black the nasal cavity leading to the trachea, leading to the two lobes of the lung that are shown at the bottom. And the goal of the mucociliary escalator is to keep the lungs free from pathogens. The way that this is done is that the cells within the trachea contain cilia, which are these little black lines extending from these cells here. And the role of the cilia is to move the mucus, which is shown in green, up and out of the trachea. Now, In individuals who have cystic fibrosis, what we may find is that this mucus may be so thick that it cannot be moved up and out of the lungs. And so the mucociliary escalator is in part inhibited in its function in that case. We can also find diseases where these cilia do not beat, and so the mucus is not being moved up and out of the trachea as they should be by the cilia. It's important to note here, too, that mucus in its own right is a physical barrier. Because of its thick consistency, it traps microorganisms, preventing them from attaching or colonizing to host cells. We've already talked a little bit about some of the chemicals that are produced on the skin. I want to point out real quick that also sebaceous glands, which produce sebum, can provide not only moisturizer to hair follicles, but also can provide an oily barrier that prevent microorganisms from being able to colonize within hair follicles. On the right, you see the gastrointestinal tract, and many of you know that the stomach has a low pH condition caused by the release of hydrochloric stomach acids. This low acid condition can serve as a chemical barrier to inhibit the growth of microorganisms. After contents of the stomach leave into the small intestines, we do find a raise in pH as bicarbonate is being secreted into the small intestines. But the other thing to point out here is that we also find other digestive molecules being produced. One of them that has an inhibitory effect on the growth of microorganisms is known as bile salts or bile. And these can actually disrupt the membranes of bacterial cells and so can form another type of chemical barrier. Do not forget that we've also talked about other chemical barriers that can be created in the human body, including these antimicrobial peptides. Some are found on the skin, and some are found in secretions, such as either vaginal secretions or tears. Even in saliva, we have lysozyme as, again, a chemical barrier against infection. And then the bacteria that are resident to the large intestine can also provide antimicrobials or conditions that might be inhospitable for the growth of other potentially pathogenic microorganisms. Now, obviously, there are times when these first lines of defense are breached. For example, a scrape or a cut can lead to entry of microorganisms underneath the skin, and this is known as the parenteral route of entry. Similarly, removal of microorganisms with broad-spectrum antibiotics can reduce diversity and create free space and an abundance of nutrients that allow for potential pathogens to colonize. These opportunistic infections may end up being the cause of superinfections. And so in the next video, we're going to begin to dissect how does the rest of the innate immune system, particularly that second line of defense, begin to protect against infection in these two cases.